Very interesting reaction is the decarboxylation reaction of beta-carbonyl carboxylic acids. Here's an example. If we start off with a ketone that is in the beta position of an ester, we can do one of these decarboxylation reactions. As a reminder, when you have an ester, the first carbon is called the alpha carbon. The next carbon is the beta carbon. And so we can call this a beta ketoester. These types of compounds, when you treat them with acid, so sulfuric acid, for example, and water, and heat it, the first thing that's going to occur is we're going to hydrolyze this ester into a carboxylic acid. So this is a reaction that we saw in the carboxylic acid chapter when we looked at acyl substitution reactions. And once you have a carboxylic acid that has a ketone or any other carbonyl at the beta position, a decarboxylation reaction will occur very readily. So this carboxylic acid, one way of looking at it, if we turn it, will look like this, facing towards the ketone. The ketone can be protonated by the carboxylic acid. That will lead to the bond between the OH group. Those electrons go in here, forming a double bond. And then this bond breaking to form a double bond here. The result of those electron movements would be an enol from the left side of this molecule. So we have a double bond and we are turning this carbonyl into an OH. So that's our enol. And then from the right side, this becomes a CO2 molecule. So this carbon is double bonded to this oxygen as well as double bonded now to this oxygen. So you have now CO2. So we have essentially removed this carboxylic acid from the right side of the ketone. The enol, as you might recall, is a tautomer of ketones. And so anytime in a mechanism you have an enol intermediate, it would be converted into the corresponding carbonyl, in this case, this ketone here. And so from this reaction, we have turned this beta ketoester into a ketone, and we have sort of eliminated this entire ester since we first turn it into a carboxylic acid and then it is decarboxylated as CO2. Note that this reaction can occur with any carboxylic acid that has a beta carbonyl. So we could have an aldehyde here, we could have another carboxylic acid, we could have an ester or any other carbonyl. A few more examples of decarboxylation reactions. This first example, a general example of a diester that is 1,3 to each other. If we heat it up with aqueous acid, so that would be a mixture between sulfuric acid and water, for example, the first thing that would happen is these two esters would be hydrolyzed into carboxylic acids. And since they are 1,3 to each other, one of these carboxylic acids would be decarboxylated. So we can, let's say, keep this carboxylic acid and eliminate this one. And so you'll end up with this product here. We'll also have CO2 and ethanol as byproducts. Ethanol came from the hydrolysis reaction. So the leaving groups on the esters become ethanol molecules. Here's a look at how this reaction occurs. So this is a 1,3 diester, dicarboxylic acid. So that would be the intermediate of this reaction after you hydrolyze the two esters. One of the carboxylic acids would be like this. The other one turns towards it so that the carbonyl can be protonated. We can reform a double bond here, break this carbon-carbon bond, form a double bond here. And the result of that would be, from the right side, an enol of a carboxylic acid. So we have a double bond and then an OH here. So that's an enol of a carboxylic acid. That's what we see here. The left side becomes a CO2 molecule. So we're forming a double bond here between the carbon and oxygen. We already have a carbon-oxygen double bond, so we end up with a CO2 molecule. The enol of the carboxylic acid, similar to the enol of a ketone, is going to be converted into the carbonyl form. So in this example, this enol becomes a carboxylic acid. In this next example, we have a carbonyl that is beta to a carboxylic acid. In general, any carbonyl will lead to a decarboxylation reaction, forming an enol initially, and then that enol becomes a ketone. The malonic ester synthesis is a synthetic strategy that sometimes we use, combining two reactions we've seen in this chapter, which is the alkylation of enolates followed by decarboxylation to make a carboxylic acid that is exactly 2 carbons longer than the alkyl halide we're using. Now, you don't have to memorize this strategy per se. Just be more familiar with the two reactions involved, which we have covered in this chapter, and then using them for a synthesis. Here's an example. If we have 1-bromobutane and we take a malonic ester, and it doesn't matter which ester we use, and we add a base such as sodium methoxide. The base would deprotonate one of these alpha hydrogens, which will lead to an alkylation of that alpha carbon. And then if we use a decarboxylation, so adding aqueous acid and heating the ester, or the diester in this case, will lead to first hydrolyzing the two esters into carboxylic acids, and one of those two esters would be removed by decarboxylation, leading to a carboxylic acid that is 2 carbons longer than the original alkyl halide. So what we did here is, imagine we remove this ester entirely through decarboxylation. The other ester becomes a carboxylic acid. It involves 2 carbons, so we have 1 carbon here, 1 carbon there, that was added to the alkyl halide that we started with. Now, we don't necessarily have to stop after one alkylation. We can do a second alkylation. Let's say remove this hydrogen with base, and we could add a methyl group in this example. And then if we do a decarboxylation, we'll once again remove one of the two esters, and the other one is converted into a carboxylic acid. We can add rings like a cyclopentane or a cyclohexane ring to the alpha carbon of a carbonyl compound or a dicarbonyl compound. So using malonic ester as an example, and adding base to it, we can make an enolate, which is already shown here. That enolate would react with a dialkyl halide. First it would react with one of the carbons with the leaving group and add this carbon chain to it. So that gives us this intermediate. With sufficient base, we can remove the other alpha hydrogen, which would once again attack this carbon here with the leaving group, and as shown in this example. And that leads to closing of the ring. So in this example, we started with an alkyl halide that had 4 carbons. Those 4 carbons are shown here, and they're both, at least the 2 ends of the dihalide are attached to the same alpha carbon, since the deprotonation occurred there twice. And then that enolate that was made in each case attacked a carbon with a leaving group, so the top one, and then later the bottom one. And so we ended up making a ring that is 5 carbons long, centered around the alpha carbon in between the 2 carbonyls. If we add aqueous acid and heat it up, we can hydrolyze these esters into a carboxylic acid. And then since they're beta to each other, one of these carboxylic acids that would result would be eliminated through CO2 or through a decarboxylation reaction. So that will leave a single carboxylic acid as shown in this product here. Notice the carboxylic acid is attached to the cyclopentane that we have here. So this carbon here is this carbon, and then let's say if we kept this ester and then eventually turn it into carboxylic acid, this carbon would be this one highlighted here. Now note that we don't necessarily have to do this second step. We could stop the reaction if we wanted to at this step. We are essentially combining a double alkylation reaction followed by a decarboxylation reaction to make this carboxylic acid, but we don't necessarily have to do that. The decarboxylation reactions of beta-carbonyl carboxylic acids is actually an important biological reaction that occurs in a few places. One of those places is the citric acid cycle in step 3 of the citric acid cycle. So that refers to going from isocitrate to this alpha keto glutarate. Notice that we have highlighted here in green carboxylate, which is the conjugate base of a carboxylic acid, and that carboxylate is beta to this alcohol. So they're on adjacent. We have a carboxylic acid, we have a carbon in between, and then we have that carbon with alcohol. In this step, NAD+, which is an oxidant that cells have available, will oxidize that alcohol into the ketone that we see here in the product. And once that ketone is formed, it's going to be beta to the carboxylic acid we had there. And so this molecule will undergo a decarboxylation right after the oxidation of that alcohol. So that is the reason why in the product we only see a hydrogen there, since the carboxylic acid is completely removed. In this question, we're going to try to use the malonic ester synthesis to prepare the following compound, which is shown here. Note that this is a carboxylic acid attached to this carbon, which has a methyl group to the right, and it has, it looks like 1, 2, 3, 4, an isobutyl chain to the left. Now, in this reaction, the question asks us to use the malonic ester synthesis to prepare the following compound. You do not need to know, at least for this class, what exactly is the malonic ester synthesis, but you should be able to use alkylation reactions combined with decarboxylation reactions to form a carboxylic acid that has certain alkyl groups on it. In the malonic ester synthesis, we generally start with a diester, such as this one, and we can use base to form an enolate of the alpha carbon. So notice these two esters are bound to this center carbon. So it looks something like this. It starts off with a carbon in the middle, and then there's an ester on the left. There's an ester in this direction. And it doesn't matter which alkyl groups are on the ester. So we're using here an ethyl group on both esters, but it doesn't really matter which ones we have. And then we're going to use base to form an enolate from the center carbon, and then we could add alkyl groups from the corresponding alkyl halides. So if we're trying to make this particular carboxylic acid, which is shown here in the skeletal structure, this is what the question asks. We just converted it into a skeletal structure. Thinking about this reaction backwards, we want to have diester. So here's a carboxylic acid that would come from an ester, and we would have another ester coming from that same carbon. And we could add this methyl group and this isobutyl group through alkylation reactions with the appropriate alkyl halides. Then if we do a decarboxylation, we can remove one of the ester groups and the other one gets turned into a carboxylic acid. So that's essentially the strategy we use. So to add a methyl group or an isobutyl group, we need the appropriate alkyl halide. It doesn't matter which one you add first. We can add the isobutyl group first. So if we add sodium methoxide and then this alkyl halide, we can add the isobutyl group attached to that center carbon, which is this one here. And if we repeat the reaction with sodium methoxide and methyl bromide, we can add now the methyl group. So that would be this methyl group here. The isobutyl group is this one here, which we already added. If we add aqueous acid and heat it, we can hydrolyze these two esters. So there's two of them shown here in parentheses. We will be converting them to carboxylic acids, and one of them would be eliminated. So at the end, we end up with a single carboxylic acid, which is attached to a carbon, which is the alpha carbon, this one, with a methyl group and an isobutyl group.